right, and the floor is yours.
Okay, thank you, Taryn. My name is Mike Harding, and I'm a plant pathologist with Alberta Agriculture. And I'm going to be spending a little bit of time here today talking about managing clubroot as part of a response to Saddle Hills reporting their first case of clubroot in the county. And I've been asked to kind of give a Clubroot 101 approach to this presentation. So I'm going to be going over some fairly basic stuff, but there should be time at the end if there's more detailed questions people have. So let's jump in here and get rolling. I'm going to talk today about clubroot. I'm going to talk about the disease on canola and other crops. I'm going to talk about what causes it. I'm going to talk a little bit about what it does to the canola plant, and then we'll spend a fair bit of time talking about what it is that we're going to do about it to try to, first of all, prevent or avoid it, and then if and when it shows up, how are we going to manage it? Okay, let's do a quick history here. Clubroot, even though it's new to Saddle Hills County, is not new to Alberta. It was first reported in some home gardens in Edmonton in the 1970s. It was present in some market gardens in the Leduc area in the early 2000s. The first reported case on canola in Alberta was in 2003, and there's been annual surveys carried out since that time, led primarily by the University of Alberta, Dr. Stephen Strelkoff. And we now have over 3,000 fields confirmed in Alberta to have clubroot. And there's obviously more than that, but that's the ones that we have confirmed symptoms showing up in the fields as part of that annual survey. So it's not just canola that's a host for this disease. So going from left to right, you have broccoli, cauliflower, cabbage, Brussels sprouts. These are all hosts for the clubroot pathogen. Again, going from left to right, kohlrabi, kale, radish, and other leafy brassica vegetables like Chinese cabbage. And there are also weeds that can get clubroot. So from left to right, we have stinkweed, shepherd's purse, and flicksweed. And so any wild mustard or brassica plant, whether it's a weed or a crop, is susceptible, potentially susceptible to clubroot. And canola being a major crop, we have lots of the host plant available across widespread acres, and that's one of the reasons why it has spread so quickly in canola. But these other crops can also and have also been reported to have clubroot in Alberta. So what causes it? If you get a chance to watch this video from, I'm not going to take time to show it here, it's about 8 minutes long, but if you go to the Canola Council's website, or just Google clubroot of canola disease cycle, you'll find this video that does a really good job of introducing what the pathogen is and what it does. And so it's really good. And I suggest that if you're interested in knowing more about clubroot, that you take the time to look at this. I'm going to just talk through the life and disease cycle of the pathogen here just for a minute, because 8 minutes is a long time for this presentation. And so let's just go quickly. I'm going to start here with the resting spore in the soil. So the pathogen that causes clubroot is called Plasmodiaphora brassicae. Now there's no test at the end, so you don't need to remember that or write it down. But you need to know that this pathogen forms very small spores that we call resting spores, and they're going to live in the soil. Okay, so that's where we're going to start. When the conditions are suitable, the spores will germinate to form a swimming zoospore. And these can swim in water films in the soil for, like a centimeter or so through the soil water. And they're going to infect young host seedlings. And they're going to insist on the root hairs and infect and feed. And then they're going to cause a secondary flush of spores. And these cause the infection that leads to the symptoms that we're going to see on the roots. So it forms a feeding structure called the plasmodium. This invades the inner cells of the root tissue, and it causes these club-shaped swellings that exist on the roots. And inside of those clubs, the pathogen then will form more resting spores. And at the end of the season, after the crop is harvested, the roots disintegrate and these resting spores are released back into the soil. Now, this resting spore is a really critical part. Understanding how this resting spore works is a critical part of management. So I want to just impress upon your minds that the pathogen can produce millions of these spores in one root, and that these spores can live for a very long time in the soil, dormant. So anywhere from 2 to 20 years in the soil, and they're not all clones. It means that there's some genetic diversity in this population of spores, so that when we bring resistant host crops in, not all of those resting spores in the soil are going to trigger the resistance reaction in the host. So there's a wide range of variability, and we call them pathotypes. Okay, so the 3 points here with resting spores, there's lots of them produced in a root. They can live for a very, very long time in the soil, and they're not genetically identical. There's different pathotypes that can kind of overcome the resistance that we deploy in the host canola crops. So these are the 3 things that make this disease special. It's not like other diseases like blackleg, and it's very, very difficult to manage. And so we're going to talk a little bit more here in a bit about what is it that we do about this resting spore issue. So I wanted to take a little bit more time to talk about these resting spores because they're so important. So here's a micrograph that's showing some resting spores. So this is looking under a microscope. And you can see here a little resting spore. These are generally 3 to 5 microns in diameter. So to give you an idea of how big that is, a human hair is about 70 microns. So you could potentially put 15 of these clubroot spores lined up across the width of a human hair. They're very, very small, and the pathogen can make millions of them in one root. So here's a scanning electron micrograph showing individual plant cells. So these are plant cells in a root, and these are the clubroot spores inside the cell. So you can imagine that if you have a plant root that has thousands to tens of thousands of cells, and many of them have thousands to tens of thousands of resting spores, that's a lot of disease potential that's getting released just from one root. Okay, so this is what makes the disease challenging and special and difficult to manage. So what are some of the conditions that are going to lead to clubroot showing up in our fields? So first of all, it's going to have to be introduced because it's not endemic to Alberta. So it's going to have to be introduced to the field. It's going to have to be introduced at a level that it can initiate disease. That's usually about 1,000 to 10,000 resting spores per gram of soil. And then once it's there, it's going to be favored by the presence of a host crop. So it has to have something that it can infect. And then warm temperatures. So we're talking about soil temperatures kind of above that 14 degrees Celsius. Acidic soils, so usually pH is less than 7, favor the disease. It needs moisture for those swimming spores, and it needs about 6 to 8 weeks of infection or incubation time before we start to see symptoms. And so you can see symptoms below ground in the form of these root galls that I'm showing in this photograph here on the right, but sometimes you can see above ground symptoms as well. Now, if you have a serious infection, you're always going to see the clubroot symptoms on the roots. You may or may not see any symptoms above ground. We'll take a look at those in just a second. I want to point out one thing though. Keep in mind that what I've said here about temperature and soil and moisture, the disease is favored. Okay, that doesn't mean it can't exist. So if the soil temperature is 11 or 12 degrees, you can still get some clubroot forming. If the pH is 7.5, you can still get some clubroot forming. If the soil's got a little bit of moisture, but it's dry, you can still see a little bit. So the environment drives the severity of the disease, but it doesn't necessarily always prevent it from occurring. So if you have high pH soils, it doesn't mean you're immune from clubroot. It just means it has a harder time getting going and causing this level of severity on a root. Okay, so if you're looking for symptoms, what are you going to look for? Okay, so here's a canola plant where we can see some yellowing of these lower leaves. They're also starting to flag a bit. And so there's an indication here that there might be something wrong. We pull up the root and look at it, and we see these swellings or clubs on the roots. And so that's a really good indication that clubroot is there and causing these symptoms. But you don't always see above ground symptoms. So for example, here's a patch of dead and dying canola plants. And this is an irrigated canola field in southern Alberta that I visited about 3 or 4 years ago. And the tops of these flowers here are above my shoulders, and I'm about 5'10". Which of these two patches has clubroot? And it's a trick question. They both had clubroot. So you don't always see the above ground symptoms. And this is why it's important to not necessarily wait till you see dead patches in your field, but to proactively scout prior to seeing above ground symptoms to try and catch it as early as possible. And we're going to confirm and talk about why that is, but I'll give you a spoiler here. The reason is that we want to keep resting spore populations as low as possible. So the earlier you can find it, the better able you'll be to keep resting spore populations low. And if you can do that, you can quite successfully manage clubroot with genetic resistance and crop rotation. But if you wait until you have, a 200 meter squared dead patch, you got a real problem that you're probably not going to be able to manage with a resistant variety in a 2-year rotation. That's just not going to work. So, and we'll talk in more detail about that, but that's the reason we want to start looking for clubroot early, even before there's above ground symptoms, is because sometimes you'll see roots like this with a healthy canopy. Okay, so this leads us to, looking for. So if I live in an area where clubroot's been reported for the first time, I'm going to want to kind of be on the lookout for it to see if it's going to show up on my farm or, in my area. And so let's talk a little bit about surveillance versus scouting. So when I get up and talk, a lot of what I talk about has to do with crop disease surveys. Now, these surveys are protocol-driven activities that usually target the whole province of Alberta and usually only involve one field visit during the season. And it's just a quick snapshot of a very small area kind of near the main entrance to the field. And so over a large geographic area, this gives us indication about what the disease is doing across the whole province. And so there's good reasons to do these surveys. And this is part of my role with the Alberta government. And we work with Dr. Strelkoff at the University of Alberta to generate the clubroot survey information. But this isn't scouting. We're not scouting for clubroot. So when we do this protocol-driven surveillance, there could be clubroot in the field and we don't find it, because we're not scouting for it, we're just following a protocol. So if you're an agronomist or a producer or a landowner, you're going to want to scout, not do surveys. And so scouting is freestyle. You just want to go find it. And so this usually targets, a field or maybe a number of fields in a farm unit. And you're going to want to visit the fields multiple times in the same season that's just part of operations. You're going to be doing that. And so while you're there in the field, you might want to take the opportunity to look for clubroot. And so how would you do that? How would you scout for clubroot? Well, when we do the surveys, we follow a protocol, and it's listed here, and it's really not that important for this talk. I'm just letting you know this is, we go to the entrance, we pull 100 plants at the entrance, walk a W with about 20 or 30 meters in between. And if we don't find clubroot at the entrance, we just go on to the next field. Okay, that's not scouting. Scouting is where you go to the areas that you're most likely to find clubroot. Now, there's a few things we've learned about clubroot in the past. And one is that for a number of areas of Alberta, we're much more likely to find clubroot at the main entrance to the field, because usually clubroot is going to be introduced on soil. And the main entrance to the field is often that bottleneck where all of the equipment and traffic is coming in. And so lots of times that's where it shows up first. It doesn't always show up at the main entrance first, but the odds are in the favor of finding it there. So that's a good place to look. When you're at the main entrance to the field, pull up some plants and look. Second place to look is low spots. So here you can see kind of an entrance to the field where water's standing. That's going to be a good place to look. Shelter belts, fence lines, low spots. These are areas where either snow accumulates and then there's more moisture in the spring, or just if it's a low spot, it's an area where there's soil moisture for the pathogen. And so these are some examples of areas where you'd want to look proactively. Anywhere that you see snowdrifts, the last snowdrifts to melt in the field, that's a good place to start looking, or along fence lines or wooded areas or low spots. Here's an example from a field not in Alberta. I think this was from Ontario, and you can see that there's a bit of a water course here, a drainage. And you can see also that there's some prematurely ripening canola. And that is actually due to clubroot infection there. And so there's a lot of more moisture there where that water drainage was, and that's where it showed up first. So these are just some examples to get you thinking about how to scout for clubroot on your farm. Now, it doesn't hurt to just do some random scouting as well, because like I said, the earlier you find it, the better. But for sure, if you see low spots, prematurely ripening plants, areas where you don't see a lot of flowers, or areas where there's a lot of weeds and low crop establishment, for sure you're going to want to check these areas, the roots of the plants, to see if clubroot is present. It takes, like I mentioned, 6 to 8 weeks for symptoms to start showing up on the roots. So it's usually scouting before flowering, the odds are against you. And then at the end of the season, kind of right at swathing timing, that's when the disease symptoms reach their apex, and they're going to be the easiest to spot. So anytime, kind of like mid-flowering till swathing timing is a really good time to be out there if you're in the field pulling some plants. So sometimes just hopping off the swather every few rounds is a good way to not only stretch your legs, but also do some scouting. Okay, so sometimes you're going to see above ground symptoms. Can you see this line right here? So this is a photograph that I got from Dan Orchard, who used to work for the Canola Council. And this area here was the old garden as part of the farmyard. And they grew brassica vegetables in the garden, and clubroot was there. Nobody knew. And so they planted canola, and they started seeing symptoms there. So that's an example of a real kind of line across the field where you can see above ground symptoms. Here's another one kind of on the side of a hill where you just have a lot of weeds and bare ground showing. That's because the plants are dying due to really a lot of clubroot damage and disease on the roots. Okay, so random scouting and early detection is an important part of what I'm talking about here. So scheduling in scouting at, between flowering and swathing, that's a really good idea. And to target those areas like the main entrance and the low spots. But random scouting is also going to be really important for this reason. This is a slide that was produced by Dr. Mary Ruth McDonald at the University of Guelph, who's done a lot of excellent work with clubroot. And she showed here on Chinese cabbage that if you expose the roots to clubroot resting spores at 1,000 spores per gram of soil, less than 1,000, and then at 1,000, 10,000, 100,000, and 1,000,000, you get this variable amount of severity in the clubroot symptoms. And so you can see at 1,000 here, you're just starting to see these small swellings on some of the lateral roots. Whereas you start to get up to 10,000, you're seeing large galls on the main root and large swellings on the tap root, or on the lateral roots. So if you can catch clubroot symptoms at this 1,000 to 10,000, you're going to be really in good shape to manage this. If you wait until there's 1,000,000 or even 1,000,000,000 resting spores per gram of soil, well, you're in tough. That's going to be a tough situation to manage. So even though it's a bit needle in a haystack to look for this, if you can find clubroot here in this 1,000 to 10,000 by some proactive random scouting, that is really going to pay off. So now there's obviously a trade-off. You don't want to waste too much time looking for something that isn't there. But at the same time, there's probably going to be times during your normal operations that you can take 5 or 10 minutes and scout, and it's not going to be the end of the world for your, your operational situation. So It's just, I throw this out there to let you know that, boy, the more proactive scouting we can do to try and find things here, the better able we're going to be to manage this problem. So here's some examples of photographs that were provided to me by some Canola Council agronomists. And so you can see a small swelling here that if you just yanked that root up and didn't brush the dirt off, you wouldn't even see it. And here's another example of some small swellings on roots that, you know, you're really catching it early if you can find this. Okay, if you wait till you see a half an acre that's dead like this, that's going to be really hard to manage. There's so many resting spores present in that soil that it's going to take years and years and years for that population of resting spores to die off to the point where you're going to be able to really safely bring canola into a regular part of your crop rotation and not see resistance breakdown of the resistant genetics. So the earlier we can catch it, the better. Now, sometimes it's because it's a bit needle in a haystack, we just aren't going to see it early. We'll have to see it when it's above ground symptoms, and that's just life. But it doesn't mean we should just bury our heads in the sand and not do some proactive work to try and sniff this out early, because the amount of time and headache it'll save on the management side is really worth the effort. Okay, so we're going to switch gears now and talk about the management side of the clubroot issue. And so we'll start first with avoidance, because that's really the best way to manage this disease, is to not get it. Now, thankfully, we have resistant cultivars available with clubroot resistance. But that's not a silver bullet that is going to last forever if we don't practice good resistance stewardship. And so avoidance is still a really important part. Now, as part of avoidance, we should be choosing now. In fact, we probably should have been already chosen this 3 or 4 or 5 years ago when clubroot first showed up in the Peace region. Choose now to grow a resistant cultivar, because you're going to be much less likely to see these large, half-acre dead patches that will just be almost impossible to manage by starting now with a resistant cultivar. This will really help to keep resting spore populations low by starting to deploy this now. Because let me ask you the question, how do you know when clubroot has been introduced to your farm? You don't know when that happens. You might not see the evidence of clubroot being introduced until, 3, 4, 5, 6 years after it's been introduced. So you need to start now, because the risk is there that you should be starting now. The second thing is, do you remember how I mentioned those resting spores aren't all clones? They aren't all genetically identical. It means that some of them have the capacity to not, the resistance doesn't work for those pathotypes. And so the good news is that most of the spores will die off in a 2-year absence of a host crop. So if you can grow a resistant cultivar and maintain a 2-year break between canola crops in your rotation, you're going to be sitting pretty. You're going to be really able to kind of avoid a lot of bad clubroot problems. That's not going to be the case if you have a dead patch with 1 million to 1 billion resting spores per gram. We're going to talk about that in the next slide. But if you don't have clubroot, or you don't know you have it yet, this is a really, really good strategy. Now, in addition to canola, though, as a host, you remember how we talked about brassica weeds? So we don't want any host crop in there for 2 years. That means you got to control volunteers and you got to control brassica weeds. And then the fourth is you want to avoid moving clubroot infested soil onto your farm. So if you're not aware of clubroot being present, you want to be really careful about what soil comes from areas that you either don't know what the clubroot status is, or especially if you know it's coming from an area that has clubroot, you want to be really careful not to bring that soil onto your farm. And then finally, you have to be scouting for symptoms to know if this is working, okay? Now, there's a lot of great information about clubroot management on the Canola Council's website, And I'm really just reiterating a lot of what's there just in bullet point form. But if you want more details, you can go to the Canola Council's website and learn a lot more about Clubroot. Okay, so what if you already have it? It's been introduced onto my farm now. I have to manage it and deal with it. How am I going to do that? Well, you're going to do exactly the same thing. You're going to choose to grow a cultivar that has resistance to clubroot. You're going to maintain at least a 2-year break in your rotation, and it's going to require more than a 2-year break if you have really high resting spore loads in your fields. And in addition to that, there may be a notice to control issued when clubroot is confirmed that will have a rotational break specified in the notice. And the purpose for that is exactly this, that you have to give it time for enough resting spores to die that you can bring resistant genetics back in and not have them break down. So these resistance breaking pathotypes that I'm not going to have time to talk about today, this is the reason why this is a risk. So if you don't know if clubroot's present, or if you know that all the evidence is that you don't have it, then resistant genetics with a 2-year break, that's great. But once you've got a dead patch, you got a new situation now that a 2-year break's not going to work. And so that's where things change once you find it. The second thing all the rest is the same, except that the second thing that's different is now you know you have clubroot in the soil, and so you have to employ some biosafety practices to prevent it from moving to other parts of your operation. So minimum field operations, sanitization of equipment coming from known infested fields, and perhaps establishing a clean exit for fields that are known to have an infested area. So what that means is, let's say your main entrance has a whole bunch of clubroot in it. You probably don't want to exit the field from the worstly contaminated or polluted part of the field. You may want to establish a clean exit so that you can leave and not take the clubroot with you, or at least make it much less likely. Okay, so we're going to need to talk a little bit about this biosafety piece, because it's pretty important, but it's really challenging to implement. And so we've got to be practical and realistic about what we can do, but at the same time, there isn't any other way to prevent clubroot from moving. So we're going to talk about some principles of biosafety, and these include avoidance, so to prevent the introduction, containment, which means to prevent the spread once it's there, and we'll talk a little bit about planning, because I think this is probably the most important part of this. We talk a lot about avoidance and biocontainment, and usually doing this through things like sanitization or cleaning of equipment, but this is probably the most important part, where we just talk about what it is that we are going to be able to do. What's practical for me to do on my farm that's going to get me ahead of this rather than just continually falling behind. Okay, so avoidance is a biosafety practice where we don't want to introduce the problem. And so if you don't have it, you're going to want to do what you can to make sure that you don't bring any soil onto your farm that's contaminated with clubroot. So it's pretty simple in theory. In practice, it can be quite challenging because there's a lot of vehicles coming on and off farms, whether they're your own or consultants or contracted services or pipelining or construction or oil and gas expert. I mean, the list goes on and on and on and on and on, and these can all potentially be risks. So how do you do this avoidance piece? Well, we're going to talk about making a plan here in a bit, because that's really the way to do it. You may not be able to be perfect at this, but we should do the best we can to try and minimize the risks. Okay, the second is preventing spread. So if I have an issue, a health issue, I don't want to spread that around uncontrolled. I don't want to be this guy that I'm showing here in the picture. That's not a good friend or a good neighbor. So we want to do our best to prevent the spread. And so that's part of the biocontainment piece, is that we don't want to spread things unnecessarily. And how is clubroot spreading or introduced to a field? it moves with soil. The club pathogen doesn't generally, I mean, it doesn't have an above ground phase to the life cycle. It's under the ground for 100% of its life. So it moves with soil. So however soil moves, that's how it moves. On equipment, water erosion, wind erosion, any way that soil moves, that's how it's moving. And so the question I think that is going to occur to a lot of people is, if I have a little bit of mud on a vehicle, is this really a risk? So there was some work done by my predecessor, Dr. Ron Howard, and he showed that a cultivator that was this size had around 100 pounds of soil on it after it had finished operation in about a quarter section. So after they scraped all the loose dirt off the tires and shanks, and this field wasn't particularly muddy, but it had enough moisture in it that soil stuck to some things. So 100 pounds. So if you have 100 pounds of soil and you have, say, 100,000 to 1 million resting spores per gram of soil, that's only 45 trillion spores that you have on that piece of equipment. So is that a risk? Obviously, that's a risk. That's a huge amount of resting spores on that piece of equipment. So how much soil could there be caught up on a four-wheel drive tractor? Well, on this machine, as you see it here, when the soil was scraped off of it, it was 600 pounds. So yes, there's a lot of soil moving around on farm equipment, and this is a risk. And so what are we going to do about this? Well, what's the best way to prevent the spread of clubroot? It's the only way to prevent the spread of clubroot, and that is to prevent soil from moving from equipment or in other ways from one field to another. So if there was another option or a better option, I would be glad to tell it, but there isn't. This is really the only way to prevent the spread. Okay, so this leads to the next question, which is, well, this probably isn't the next question, but this is the next slide here. When I'm talking about preventing movement, I'm talking about sanitization of equipment. And this means rough cleaning, which is scraping off or knocking off as much dirt as we can, fine cleaning with pressurized water or compressed air, and then finally disinfection. Okay, so here's the practical problem. We just don't have time to do this for every single field that we're going to leave, especially during a busy seeding window, short seeding window, or short harvest window. I don't have time to do all this. What am I going to do about it? Okay, well, it's okay if you can't do everything, but it's not okay to be this guy and do nothing. So this is where the planning part comes into it. I'm not telling you have to wash every piece of equipment every time you're coming in and out of a field, but I'm telling you that if you just say, I don't have time for this, I'm not going to do anything, you're making a mistake. So you need to decide what you can do. What do you have time to do? I'll give you an example. What if I had time to kick the dirt off the openers before I left the field, and that removed 25% of the soil? You just reduced your risk by 25%. What if I had time to knock the loose dirt off of tractor and equipment with a long-handled scraper, and I reduced another 25%? And then what if I exited in a clean exit, and that reduced my risk by another 25%? Okay, you just reduced 75% of your risk in probably a 30-minute exercise leaving a field. And if you knew that field had clubroot, would you be willing to invest 30 minutes out of your day to not spread it to another field? So I would be, because I know how difficult clubroot is to manage once it's introduced, and that the best practice is to avoid it. Now, you may not be able to do this. This is just one example, but decide now, make a plan for how you're going to prevent clubroot from being introduced, either through your own activities or activities of others coming onto your farm. Make a plan that's realistic, and also make a plan to contain if it does show up on my farm. What level of sanitation or biosafety am I going to go to make sure that it stays where it is? And then implement that plan. Now, there may, again, when clubroot is identified, there may be a notice to control, and there may be some things that you're going to have to do as part of that plan. But it's a great idea to figure some of this stuff out beforehand so that you're ready to go and it's not a surprise and you've decided to do it, not somebody's telling you to do it. So I would really encourage you now to come up with a bit of a plan, and it could be scratched on a napkin, It can be just taking some time while you're driving or have some downtime and think about, what will I do now as a biosafety plan for my farm with respect to Clubroot. Now, the good news is that biosafety planning isn't just effective against Clubroot. It's effective against any weed, so prohibited noxious weed, invasive insect, anything that travels with soil. You're going to get traction with sanitation and biosafety for all of those other things as well that could potentially show up. Okay, so what have we talked about today? We've talked about the fact that the Clubroot pathogen is special. It's unique and it's difficult to manage because of those resting spores. There's so many of them. They can be dormant for so many years in the soil, and they have a lot of genetic variability that makes them able to overcome the resistance that we deploy in our canola crops. The second is that the canola survey isn't scouting. So when you see results of the survey and you don't see a red spot near your farm, that doesn't mean that there's no Clubroot there. It does not tell us where Clubroot is hiding. It just tells us that when we follow this protocol in this number of fields in the province, this is the result we get. So proactive scouting on your farm can make a big difference. First of all, to understand if you can find evidence of it being there, and second of all, to find out how well your practices and avoidance strategies are working. So when we're scouting, we know that the odds are in our favor of finding it when we look at entrances, low spots, areas of high traffic, shelter belts, fence lines, etc. And it's important to watch out for patches that become weedy or where canola wilts, where there's some above ground symptoms. Don't assume it's sclerotinia or blackleg or a drowned out spot. You need to go look and see if Clubroot's there. So that proactive scouting and random scouting, just anywhere in the field while you're doing your normal operations, especially during harvest time, is going to be time well spent. And then finally, when it comes to management, follow the management practices that are outlined in the various sources that I've shared with you today, and you can get them from other places as well. But really think about making a plan. So what is going to be your biosafety plan, and how are you going to execute that to prevent introduction and to prevent unnecessary spread specifically on your farm? What is it that you're capable of and prepared to do to try and manage this problem? Okay, so I hope I've shared some things today that have been helpful. And if we have time and ability to go over, discuss any questions, I'd be happy to try to do that. And thanks very much for your attention.
Thanks for presenting. I actually have a question for you.
Sure.
Where would you suggest producers send their Clubroot samples if they do find something they think is suspicious?
Well, that's a great question. So if you need a very quick turnaround, so you want to know right away, then you should send it to a commercial testing lab. And I think there's a list on the Canola Council's website of all the testing labs across Western Canada. I am aware of SGS BioVision and 2020 Seed Labs as two of the primary Clubroot testing facilities in the province of Alberta. And so you'd want to send it to a commercial lab that can give you a very quick turnaround. Now, if you don't need a quick turnaround, you could certainly talk to the ag fieldman in the county because they have access to the plant health lab in Edmonton, and those tests are free of charge. But it could take 2 to 3 to 4 weeks depending on the queue that exists there. So did that answer the question?
Yeah, it did. Thank you.
I see Norm's hand is up.
Would it be okay if I asked a question?
Yeah, go for it, Norm.
So when you talk about finding something that you realize is a heavily infested patch, what type of treatments would there be for a patch? So potentially taking it out of production, treating it separately from the field. What could a producer do about something like that?
That's a great question, Norm, and we could do a whole 1 hour presentation on the details of how to do patch management. So again, if you go to the Canola Council's website, that's a great repository for a lot of this information. But I'll give you a few examples here quickly. So one is, we know that there are things called bait crops or trap crops. That's probably not really the correct word to use, but some plants will wake up the resting spores in the soil, but then the resting spores can't infect. So you can increase the reduction in resting spore viability by planting these crops that kind of wake up the spores. There's been a fair bit of work done on this over the last 5 years, and some of the results are a little inconsistent, but there's definitely evidence that certain things, certain types of crops, perennial ryegrasses and things, can help to get the resting spores to die off a little quicker. So that's one. Now, the second part of seeding a sod-forming grass over a canola patch is that it holds the soil in place so that it doesn't move around. So seeding a sod-forming grass over a clubroot patch is a good way to hold the soil in place and accelerate the resting spores waking up and dying. There's additional things like soil amendments. So if you have low pH soils and you can add lime to those soils and increase the pH, sometimes that will suppress the amount of clubroot severity and can keep the resting spore populations low. So lime, wood ash, etc. And then there's also soil fumigation has been tried in some instances. So this is where really basically you're kind of nuking the soil. You're killing just about everything that's in it with some of these fumigation exercises. So it can be quite dangerous, and you have to put a impermeable film over to keep the fumigant in the soil. So it's a lot of work. It's kind of dangerous work, but it's an alternative to try to kill those resting spores in the soil. So that's an example of some of the patch management techniques that can be utilized. Oh, soil solarization is another one that's been investigated, where you just put a clear plastic film over the soil and let the temperature, so you get solar radiation going through the plastic, but the heat can't come out of the soil. So you essentially heat up the soil to temperatures high enough that over a long enough period of time, the clubroot spores will die. So those are examples of some things. So that's the good news, is that there is some ways to kind of poke at this problem in a patch management way to try and reduce the resting spores in the soil. That's the good news. The bad news is that these resting spores are incredibly durable. They live for a really long time, and they're very, very hard to kill. And as a result of that, these treatments aren't going to be 100% effective. So again, it's incrementally adding to our ability to manage, but you can't get rid of clubroot from a field using fumigation or liming or other soil amendments or solarization. But you can certainly decrease the amount of viable spores that are present using these. So it is very valuable, and it's really good to think about because a lot of these patch management treatments are quite expensive per acre and require a lot of input, either in labor or in money. So to do it for a whole quarter section, it just isn't reasonable. But if you had a small patch, it certainly would be worth thinking about as part of your management, your proactive management of clubroot. I don't know, Norm, what do you think about that question? What do you think?
I think you covered it very well. I can't think of anything that I would be adding.
Wonderful. Thank you for doing your presentation for us tonight. I appreciate it. Let me just turn the recording off here.